Okay, good. Yeah, so this Zwitterion, but the point I was making with this Zwitterion is that that only is applicable to the N and the C terminus, right? It has nothing to do with that R group. Okay, so when he asks you the question, what is the charge on the amino acid at a pH below the PI, so below the amino acids isoelectric point, okay, you have to be looking at either the N or the C terminus. And so in this case, the cheat sheet will be, if your PI is below pH, you're more positive. If your PI is above the pH, you're more negative. And that makes sense, okay? In terms of hydrogens missing or adding extra, etc. Okay, the next thing, any questions on PI? Isoelectric.
Does that mean when PI equals pH, we have a Zwitterion?
Bing, bing, bing. Okay, because we're neutral. Exactly. Brilliant. I guess I could write that down too. If our PI equals the pH, then we have plus and minus, and I'll put CN, right? So CN terminus. Okay, the next thing you need to know is you have to be able to describe what is happening in a dipeptide bond formation. So first of all, people are going to get thrown off by dipeptide. They're going to think this is something new. It's not. Di means two, so you have two peptide bonds coming together. When we have two peptide bonds coming together, what happens? We're connecting what to what?
The C terminus to the N terminus.
And what is lost?
Water.
What is released?
Water.
Exactly. Okay, that is what you need to know. How he's going to confuse you, he won't say N terminus, he won't say C terminus. He's going to say carboxyl. He's going to say amine. He might even say NH3. We know anytime there's an N there, that's amine, okay? So look for NH3, look for COO minus, okay? Because carboxylic acid, carboxys, we know that that's COOH. And if we're losing water, then it's going to be a COO minus, okay? So things to look for in this question, COO minus, NH3, okay? And you need to know that they're going to have water removed. That's what's happening when you join two peptide bonds, two peptides, okay? Two amino acids, dipeptide. All right, any questions on that?
With the amino group, would it be going from NH3 positive to NH2 with a zero net charge?
So in that case, right, if I give you, let's see, yes, but he won't tell you what it's going to go to, but you're correct in that, what it will be. You're correct in what that NH is going to turn into. It'll be an NH2, right, when it connects. But in the question, he's not going to give you that. He's just going to say NH3 is going to bind to blah, and then blah binds with blah, and it releases blah. So you don't have to do the mathematical configuration in terms of what's happening with the hydrogens. You just need to be able to identify the N terminus is NH3, the C is the carboxyl, and then what it releases. So very, very superficial. Does that help, Riley?
Yes, thank you.
Okay. All right, so the next.
Can I ask a quick question?
Yes, absolutely.
This might be skipping ahead a bit, but is there anything special we need to know about peptide bonds forming when you make like a polypeptide, in terms of like when you go through the whole translation transcription process and it's getting made in the ribosome?
Okay, no, because that would really be getting into like the genetic side / really into that DNA, and so no, you don't have to know about that kind of stuff. That's a little bit too descriptive.
Thank you.
Yeah, the only thing that you need to know is, I guess I should also ask, what kind of reaction is taking place when you make a dipeptide bond?
Condensation reaction.
Okay, that's the extent of what you need to know in terms of this kind of stuff. Who asked that question?
Me. I'm
Sarah.
over here.
Okay, well, yeah, Sarah, that's the extent that you'd have to know. You just have to know the type of reaction that's undergoing there. So condensation, you need to know what two things are connecting and then what's leaving. Okay, that's about it.
Thank you.
All right, next question. What is the process? Now we're gonna kind of switch gears a little bit. What is the process of altering the shape of a protein without breaking the amide bond that forms the primary structure? Again, what is the process where we alter the shape of the protein without breaking the amide bond that form the primary structure?
Is it the denaturation?
Okay, so if I give you options of oxidation, denaturation, inhibition, or hydrolysis, would you be confident in saying that it is denaturation?
Yes.
Okay. What does it mean to be denatured?
It's like the unfolding of the protein. So the backbone is still the same, but all those big complicated folds, like the hydrogen bonds, are breaking apart, right?
Exactly. And what does that mean for the enzyme itself?
It loses functionality.
Loses functionality, okay? So there might be a follow-up question. When an enzyme or a protein is denatured, what does that mean in terms of its functionality? Okay? So when we start when a protein or an enzyme is getting denatured, that means it's pretty much dissolving. It's no longer able to function. Another way to think of denaturing is when we put a particular protein in a different nature, a different environment. Ways that we can denature proteins: change in pH, change in temperature. Those two things are the key factors for denaturing proteins, okay? So those I want you to also be aware of. How can we denature a protein? Okay. Next question. What type of inhibitor binds to an enzyme but does not bind to an active site?
Allosteric.
Non-competitive.
Okay, let me write these down. Oopsies, lost a little. Okay, so let's talk about inhibitors. So, irreversible, reversible, non-compet, compet, and then I did hear allosteric. Okay, if I know how to spell allosteric, that was extremely dyslexic or whatever. There we go. Okay, so the type of inhibitor, okay, so we have an inhibitor that's gonna bind to an enzyme. So here's my enzyme, okay? But it doesn't bind to the active site. So let's just say that this is our active site, and this inhib is gonna bind somewhere here. So if you're not binding to the active site, what are you?
Is it a non-competitive inhibitor?
So why would it be non-competitive?
Because it's not competing with substrates for the active site.
Right. Okay. So let me tell you something about this since we heard about it. So a non-competitive, okay, first of all, everything wants to get into that active site. Why do we want to get into the active site? So if we weren't talking about an inhibitor in general, why would something want to get into the active site?
If I remember correctly, I don't know what type of bond it is, but I think there's something like side chains in that area poking out that can form bonds with other things in the area.
Oh, oof. Let's try this. actually, I'm going to do like this, and then I'm going to draw like, man, and then I'm going to draw like this. This is a really wonked enzyme. Kind of looks like a head. Okay, so let's just say this is my enzyme, my really weird enzyme. And let's say that this is going to be, this is the active site right here. The one that I'm gonna just like this. This is our active site, and then what would this be? Do we know? So, okay, let me ask you a question. For an enzyme to work, what's going to happen here? What's going to happen in the active site? If something attaches and fits like right here, what's going to happen to this enzyme then?
Won't it modify its shape to make sure that it's a perfect fit?
Right. So we have a couple of things. Okay. So the best way for me to draw this, let's see if I can give you a little bit of a diagram, because sometimes these inhibitors are weird to understand. Okay. So we have substrates, right? So if this is the enzyme, this guy's the enzyme, right? All of this is the enzyme. Then we can have a substrate that has a point like this and maybe whatever. It doesn't matter what the part is, but this part matches up and fits in, and then we are able to do this conformational change, and then what? We build this enzyme substrate complex, and then we can basically move on with whatever processy we're doing, right? So catalytic, or we can do a cascade and do more work, right? So the point is we need to have this substrate come in that fits perfectly, conformational change, and then we basically get a result. Okay, this would be what, if we have an inhibitor that goes in and he's just maybe, I don't know, sits over here for some reason. Okay, so allosteric, we'll get into allosteric. But if we have non-competitive inhibition, what does that mean in terms of what's happening to the enzyme?
Doesn't it change the shape of the enzyme so the substrate can't fit in the gap? Can't fit in the active site?
So, right, so a non-competitive, well, when your inhibitor really, it doesn't bind. So your non-competitive inhibitor does not bind to the active site, okay? Because it doesn't actually fit there, all right? So a non-compet would be like this. This could be your non-competitive inhibitor. I'll put non-competitive inhibitor. And he's obviously a square, and he can't fit in that site, okay? So it's not going to bind there to make that enzyme work, right? But it will still, okay, bind to an enzyme. And so I guess what I could say is it could try to fit in there because there might be a point. So if I were to draw this guy again, this square may be able to fit like this, right? He may be able to, that's a little bit of a big square. We'll draw him in blue too. He may still be able to fit in there, but does he fit perfectly? No, right? So it doesn't really bother. It's not competing for that space to go and do some sort of processing. It's just kind of there, might fit in. He could bind and might make a conformational change, but it's not going to do that willingly. It's not going to be like searching around trying to find this enzyme. It just, if it happens to be there and it happens to see the active site, then it might do it. Does that make sense? It's not actively searching, okay? So you guys are right in the fact that it may go and do some sort of conformational change, but it's not doing so aggressively, okay? And it also doesn't do it with a perfect fit. Cool? So now the other thing that I want to talk about is a competitive inhibitor, right? And remember, what's the role of inhibition? What are we trying to do?
Prevent the enzyme from being able to like make that enzyme substrate complex.
Right, right. So a non-competitive inhibitor will still bind, but you're not going to have that enzyme substrate complex formation, okay? Because it just doesn't fit. Competitive inhibition is actively searching and binding to an enzyme, okay? So so this is a non-competitive. If we drew this enzyme again, a competitive enzyme inhibitor would actually have a point, okay? And it's going to resemble the intended substrate, all right? So it's going to try and beat out that substrate so that it can prevent the processes from occurring. So a competitive inhibitor resembles the actual substrate for that enzyme, okay? It's like mimicry. And it's purposely trying to get there so substrate can't, so we don't have these cascade effects, okay? Are we all clear with the difference between non-compet and compet? Compet looks like substrate, it's aggressive. Non-compet doesn't necessarily fit, but still can bind, but it kind of just walks around, and if it happens to see something, it'll go in there. Cool?
Does the non-competitive, when it is binding, is it always going to kind of fit in that active site?
Yes.
Kind of, or is it ever going to be on a different part of the enzyme? Because I remember there was an option where it binds
allosteric
to like the back end of the
inhibition. You're talking about allosteric. Yes, so competitor and non-compet go to the active site. Now, allosteric inhibition, which is why I drew like this second hole on the enzyme, allosteric inhibitor binds to the enzyme when all the active sites on the protein, though, are slightly changed and they work less. So there's like another spot on this enzyme that can kind of go in and it will alter, okay? So this is an allosteric site, this is an active site. So let me just draw a simpler enzyme again because drawing is weird. Let's just have this be this, and then we'll have this be this. Oh. You know, why can't I ever make a triangle? For some reason, it's the weirdest thing. Okay, there we go. Okay. This is your active site, and this is an allosteric site. Okay, enzymes have both, right? So again, allosteric regulation, allosteric binding, allosteric sites, allosteric molecules, it's just letting you know that when there's an enzyme that it's another site on the enzyme that an effector in terms of regulatory or allosteric may bind, okay? So allosterics is just anything but the active site. Okay, in terms of allosteric inhibition, okay, it's some inhibitor is going to bind to that secondary site on the enzyme, and it will still alter the protein conformation, right, within the active site. So let me show you what I mean. Here's allosteric inhibition. Let's go back to black because I don't like that blue. Here's allosteric inhibition. And then I'm going to draw another enzyme because this is just what we do. Okay. Here is allosteric protein or allosteric, whatever you want to be, and he's going to fit here. Okay. When he fits there, I'm going to draw another arrow because it's just like, let's do this. And then I'm just going to erase this. Now what's going to happen is you have this binding here, okay? He fits. What then, though, is changing is this part right here. So when an allosteric site is bound to, or an allosteric inhibitor binds to the allosteric site on the enzyme, okay, so now we're bound. It changes the shape of the active site. So now we have this guy that may look like this. So he went from a triangle to a square. Okay, so before the substrate had to be a triangle, now with allosteric inhibition, we've bound to the backside pretty much of the enzyme, the opposite side. We've changed the conformational, the way that the conformational shape of our active site to now be a square. That's saying, okay, now we only can have square substrates. So all of those triangle substrates no longer work. We only can have squares now. Okay? Does that make sense? Therefore, what it's saying is the enzyme no longer can bind to that specific substrate previously. So if you had a bunch of that, it's just another way to prevent catalysis or prevent a process from happening. Okay? This one is just a little bit different.
The question you asked before, what type of inhibitor binds to an enzyme but not its active site, the answer is allosteric then?
No. So the type that does, yes. So if it doesn't bind, but in this case though, he's going to say what binds to the enzyme but not at the active site. So it should be allosteric inhibition. Okay. But he's not going to give you that. He's going to, I don't see that answer on the quiz, which is slightly concerning for me. So I think it might be a typo in that case. I'm going to have to reach out to him. But the type, this was saying what type of inhibitor binds to an enzyme. Yeah, it should be an allosteric inhibitor, right? The options only say reversible, irreversible, non-compet, or compet. But I also think in this case, I don't know if he's trying just to get you to say non-compet, but.
Well, it's interesting because like I've been going through the reading and stuff and I don't remember it ever saying anything about allosteric inhibitor, and it didn't say anything about it in the like mastering either. So I'm just wondering if.
Yeah, so technically this should be allosteric, right? When it doesn't bind to the active site, that is what an allosteric, so that's why Riley said allosteric. In this question though, I think what he's looking for is non-competitive inhibition, because if it's not going and searching for your active site, do you see where we're at with that? So I don't like that question, and I'm gonna have to edit that, or at least email him. Hopefully he'll email it, but he probably is looking for a non-competitive inhibitor for that question. Okay, but technically it should be allosteric inhibition. Okay, right. What is the strongest type of interaction between two cysteine amino acid side chains in a protein?
Disulfide?
Cool. Any questions on that?
Can you say that question one more time?
What is the strongest type of interaction between two cysteine amino acid side chains? So you need to know about cysteine and what cysteine looks like in terms of the amino acid. And you, based on what's happening in that amino acid, you'll be able to tell me what's the strongest type of interaction there.
What was the strongest interaction again? Sorry, I missed it.
Okay, so what does cysteine, the amino acid look like in cysteine? What does the amino acid cysteine look like? Draw that out. Anybody know?
It has an SH group on it, doesn't it?
Right. So here's cysteine. So if I were you, the first thing that I would do is I would make sure that we can draw out the backbone of the amino acid. So let's do that. We have our NH, what? Is it H? We have NH2, right? Everybody agree? And then what? Nobody knows the backbone of an amino acid? I'm scared.
Carboxyl group, another hydrogen.
Very beautiful. Okay, so this would be the R group. So for cysteine, oh, there's an E here. This has an SH bond, okay? This right here is SH. Out of all the bonds that are happening here, we can only identify hydrogen, right? We can clearly see that there's hydrogen bonding here. There might be other intermolecular forces occurring, but this strongest bond is going to be the fact that we have sulfur, okay? So the disulfide bond is going to be the strongest if we had two amino acids, two cysteine amino acids together. That disulfide bond is going to be the strongest, okay? So 2X cysteines together is going to be a disulfide, okay? And that's your strongest. Alrighty. What is the smallest type of RNA?
Would it be tRNA?
So what are our three options?
tRNA,
mRNA.
mRNA.
Beautiful. So ribosomal, messenger, transfer. Okay, so out of these, which one is the smallest?
Would it be the rRNA?
So ribosomal?
Yeah.
So let's think about that, right? So our messenger RNA does what?
It brings genetic information, essentially a copy of the DNA, out from the nucleus out into the cytoplasm to be made into a protein.
Okay, so we know that that guy has to be big because he's reading that. He's reading that RNA, okay? So he's pretty big. Then what do we have? After messenger, then what do we have? What would be the next step?
The
A ribosome.
So it goes from mRNA to rRNA? Is that what you're saying?
Well, that's where I'm a little confused with this, because I remember the textbook said that the ribosome was composed of lots of proteins and rRNA. I would assume the rRNA would be the smallest because it's a bunch of pieces of it that make up the ribosome.
Okay, so let's think about that. Let's backtrack. So mRNA is going to account for about 5% of all the RNA in the cell, okay? It is going to basically carry, all it is both base and sequence and size. It's only about 5%, okay? All it carries is just the complementary code copied from DNA, all right? So we know that that can't be the smallest because it's carrying the copy of RNA from that, carrying the copied genetic code from DNA. And if we look at DNA, DNA is very large. There's no way that this can be mRNA, okay? We all agree with that. The next part that we're confused about is the rRNA. So our rRNA is found in the ribosome, okay? But it accounts for 80% of the total RNA present in that cell. So because ribosomes are the largest subunit, okay, could we say then, if rRNA is found in that ribosome, it is pretty much is the ribosome, right? How can we say that that's the smallest if it's making up the ribosome?
I guess in my head, I thought of it as like a bunch of individual small pieces
No.
making up the but so it's basically like big chunks of it.
Big thing. Yep. It's 80% of that pretty much. Okay.
All right, thank you.
Yes. Okay. So they do, I think what you're going through is even on the next level, because there are different types of rRNAs that are small and then are large, but we're just looking across the board very generically at these different types of RNA. And I just want you to think of it as, in general, what is RNA? You don't have to think about it in any bigger than that or any smaller than that. Because technically you are right. There are bits and pieces of RNA that are very small and then very large, but in general, it's still RNA. And so that would be a moot point. You feel me?
Yeah, thank you.
You're welcome. So that would only leave us with tRNA. So tRNA is the smallest of the 3 RNA, not only because it only has about 75 or 95 nucleotides, but all it does is translation. Okay? It just transfers the amino acids during protein synthesis. While it's a very important role, it's a very simple role. Therefore, it's the smallest. Okay? So it doesn't build anything else other than that. It's just translating it, right? It has one duty. That's it. So it has to be very small. It's very precise, and it just translates the mRNA. Okay? Any other questions? So this guy is the smallest. Okay? All right, next question. What is the process by which DNA makes a copy of itself when the cell divides? What is that process called?
Replication or mitosis?
Okay, so mitosis is dealing with the cells, right? The cells actually replicating. Think smaller. So what was your first thing?
Replication.
Right. So when DNA makes copies of itself, when it divides, I would consider that replication. How is that different than duplication? Is it? Do we know what duplication is? It means nothing. Don't fall for it. It's a trap. Okay? DNA replication is just when DNA makes a copy of itself. Okay? All right. The next thing that you need to know, I'm not going to draw this out, but I'm going to write a note here. You need to know the structure of ATP. Okay? You also should know the structure of ADP. Okay? What is the dead giveaway for the structure of ATP?
The 3 phosphate groups.
Good. Okay. That's all you need to focus about. Okay. Look for those phosphate groups. All right. I will tell you there will be images on this exam. So that should be very easy for you to figure out. The next thing though that I will tell you is you need to look for the phosphates. But then I also want you to look here. What are on these carbons that makes it ATP versus ADP?
The hydroxyl group, yeah.
So what goes here?
NOH.
And what goes here?
NOH for for the RNA.
So another OH goes here, or is it H?
No, it would be OH because it's the ribose.
Right. So OH, right? So what you need to do, okay, on these exams, you can't just look for 3 phosphates. You also need to take a look at the sugar, okay, because he will trick you. Alrighty? Please, 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 please. I know the first glance is to always look for those 3 phosphates. I'm only telling you you're going to be able to eliminate 2 of those, okay? You need to be able to remember the sugar component. Cool.
And
All
Liz,
right.
just to clarify, sorry, the OH again, which one is that? Like versus an H?
Okay, so then if we just had an H on that, does that change anything?
Yeah, that I just forget what it's called, but I know it's a completely different thing.
Okay, does anybody know?
Do you mean it's
deoxyribose has 2 hydrogens, ribose has a DOH group.
Okay.
There you go. So I think the way that he'll, so in ATP and ADP, we don't change the base, the sugar base. So the sugar base doesn't change here. The only time that this sugar base will change is when we're talking into DNA, RNA, or I should say deoxyribose, yeah, and then RNA, right? So this will have how many OHs on it? This will have OH, right? And then this has, or this deoxy has OHH because it's deoxy, and then this is OHOH. Okay. So deoxyribose sugar, I should just say this will just have these here, whereas this will be here. Okay. Does that help you?
So is there only deoxyribose in the DNA, or like in the DNA actually?
Yeah.
Like otherwise it's always a ribose.
Pretty much. Yeah, what you'll be doing, you won't, I mean, you could talk about sugars, but you won't really need to do that for this exam or this class, but yeah. Good question. Okay. All right, next question. But yeah, he'll trick you with that. You need to know the 3 stages of translation. Yes.
So what's the I guess I'm just confused of like, what's the full difference between ATP and ADP?
The only thing that's different is the amount of phosphates. But what I'm trying to tell you is he will trick you by changing the sugar base.
Gotcha.
Yes.
Okay.
So you need to make sure that with ATP and DP, you're still having a double OH there.
Perfect.
Okay?
Thank you.
Yeah. So that's the big difference there. Really, it's just the phosphate change, but he'll throw you for a loop if you don't know what the sugar base looks like. Okay. Now we need to talk about translation. All right. And there's 3 steps in translation. Okay. So the ones that you need to know is the first step is going to be what?
Initiation.
Right, it's initiation. And why? Because we can't start reading or writing, because translate is basically to write. So transcript, we read it, translate, we write it, okay? Or translation, yeah. So you need to think about, what are we doing here? We need to initiate contact. That's always going to be the first rule. Then what do we do?
Elongation.
And what happens with elongation? What are we doing? What's the role?
We're adding nucleotides onto the mRNA strand that we're creating that are complementary to the template DNA strand.
Exactly, okay? All we're doing here is adding on, okay? That's exactly what it means to elongate. We're adding on and we're building, okay? The last thing is, well, we can't do that forever, right? So we need to terminate, okay? So those are the 3 things. Mutation is not one of them. Now, mutation can occur during translation, but that doesn't mean it's a step of translation. It's more like a misstep of translation, Okay, so if something happens wrong during the translation process, then we would get a mutation. So a silent mutation, a frameshift mutation, things like that, stuff that you didn't need to know about, right? Mutation is not part of the natural process. It's not intended, okay? The mRNA, tRNA, RNA, they don't go in there thinking that they want a mutation to occur, all right? While not all mutations are bad or lethal, that's still just not a natural step of translation. Okay, any questions on that? Cool. How are the correct amino acids brought to the ribosome for protein synthesis? Let me repeat. How are the correct amino acids brought to the ribosome for protein synthesis?
Codons pairing with anticodons with tRNA.
Yes. Okay. Dead giveaway here. You need to know tRNA is being used. Okay. He is responsible for making them, reading what mRNA did, taking those 3 base pairs, translating it into the amino acid, and then we can move from there. Okay, as far as codons, stop codons, and things like that, I don't know if you read about those, but you're not going to actually be doing that for this exam. Okay, so you don't need to know UAA, UGA, you don't need to know start and stop codons. Okay, but you just are going to have to know the word codon or anticodon loop or things like that. Okay, all right. Oh, my mouse is not letting me. Hold on a second. There we go.
So is the answer to that question codon or anticodon, or is it both?
So the codon is what? It's coding for that amino acid, right? Like it's going to start the chain, the identification. It's going to start the chain of amino acids to be recognized. So what's happening is the tRNA is bringing it to that start codon from the mRNA, okay? And then we'll start the loop. Okay, because codons, like I said, they're basically like switches. And so if we have to have a switch that turns on, so we have a start switch, okay? So then tRNA comes, grabs things, here's mRNA. He reads things from mRNA, this we'll just call tRNA. Start codon, and then we can 1, 2, 3, 1, 2, 3, and start reading it. Start reading all of these things, okay? Really? That's terrible. Okay, which of the following best describes the primary function of mRNA in a cell? What is the function of mRNA? We just talked about this. What is its function? What does mRNA do?
Just transcription, right?
Right. So what's the key word that we should be looking for? Keywords.
Transcribe.
Transcribe. Okay. Let's talk about what is the difference in how many hydrogen bonds take place or are in a G and a C bond?
3.
Okay, and how many are with A and T?
2.
Okay, you need to know those things, okay? That was, I think, also on like pretty much a problem homework or a homework that you had to do. Okay. Where do we find hydrogen bonds?
Between the nucleobases and DNA.
DNA, okay? Simple terms, hydrogen bonds take place in DNA, okay? That is where they will be found. We don't give a crap about hydrogen bonds anywhere else except for in DNA, okay? So please note that. DNA, 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 DNA. The next thing is, oh great, he's bringing in coronavirus. Okay. Did you guys read about COVID-19 vaccines in his assignments or readings?
Is it just how it has to do with like one of them's got like the mRNA type of vaccine, which is unique to what we normally use for vaccines?
Yes. Keyword mRNA. Okay. Another thing that you need to think about is, what is an alpha helix?
It's a secondary structure.
It's what?
It's a secondary structure for proteins.
Okay, what's unique about it?
Is it just that like every fourth group is like hydrogen bonded together, and that's what makes the right-handed corkscrew?
Okay, so if he were to ask you certain characteristics of an alpha helix, or say which one of them is not a characteristic of an alpha helix, do you know enough about the alpha helix to tell me, to be able to tell me or identify something that's not a characteristic of it? So, I don't like the way that's questioned. Okay. Let's just go through this question. So the questions that say which is not a characteristic of an alpha helix. The R group of the amino acid extend inward toward the core of the helix. Yes or no?
No.
So R group,
the non-polar ones do.
Okay.
Right.
So then let's look to the next one. Just hold that thought. The NH and the COO bonds point along the axes of the helix in opposite directions for an alpha helix. The NH and the COO bonds point along the axes of the helix in opposite directions. Do we know what that's asking?
I don't really.
Okay, let's go through these. Let's see if I can draw. I wonder if I can pull a picture. So remember when he made you draw the DNA for your lab? Okay. So let's do, I think this is still working good. Okay, so alpha helix, okay? We know that this is secondary structure. Okay. And we also know that it's a right-handed helix. Okay. This right here then means that in the backbone, okay, so every backbone NH group, so I'm going to say NH backbone group, that the hydrogen bonds to the backbone of the CO group, okay, of the amino acid. So the NH backbone of this in every backbone of the NH group, the hydrogen bonds to the backbone of the COO group of the amino acid. Okay, so then this goes for the amino acid. So that's a characteristic. Okay, the NH hydrogen bonds to the COO bonds of the amino. okay? The questions on your exam is saying, okay, that the NH and the COO bonds point along the axes of helix in opposite directions. Are there 36 amino acids in each, 3.6 amino acids in each turn of the helix? Or the COO group of one amino acid is hydrogen bonded to an NH group, 4 amino acid residues farther along the chain. Okay, this one is always going to be 3 to 4 residues. Okay, usually it's 4 though. So these are characteristics of an alpha helix. All right, these are the things that you need to know. Do we have anything on there that talks about an R group?
If I remember correctly, I think it's just that the side chains typically point outwards.
Yes, and in this he says they extend inward. Okay, so the big takeaway here, he's saying the R group of the amino acid extend inward toward the core of the helix. Is that true or is that false? In alpha helix.
So that would be false, right? Because the nonpolar, what I was talking about, happens during tertiary formation. Right. Okay.
Right. So everything else is moot. We need to focus on the R groups for the alpha helix. Okay. That is not a characteristic of alpha helices. Okay. Is the R group. This, I just put new batteries in my mouse and it's literally sitting here saying connected, not connected, connected. Okay. Any questions on that particular question? Any questions on that question? Okay.
A question with the 3 to 4 residues. What's happening every 3 to 4 residues?
That's just how far apart those bonds are. Like they're going to go every 3 to 4. So in between, then they'll do another one.
All right. And so those do run in opposite directions.
Mm-hmm.
Okay.
Okay. The biggest takeaway, no R group involvement. All right. The next thing that you're going to be given comes identically from the practice exam that's in the file. It's going to give you a picture of valine saying, consider the following forms of the amino acid valine. Which statement concerning these structures is false? Okay. Then it's going to show you 4 images of what valine could be. And it'll say, this one represents a zwitterion. This one represents at pH, blood physiological pH. This one is basic. This one is acidic. Okay. Key takeaway here, you need to be able to identify what makes an amino acid basic, acidic, makes it a zwitterion, okay, or otherwise makes it like neutral or like what it would be at blood pH. Okay. You're going to be looking at your N groups, you're going to be looking at your C groups, and you will also be looking at the R group here, okay, for this particular question, right? But here, actually, for valine, the R groups are all going to be the same. So the only thing that we need to look for, as I redact because I looked at the question, you only need to pay attention to the N terminus and the C terminus. That's what you should be paying attention to in terms of trying to find the correct statement, all right? Because your R groups are never going to change in valine. If they did change, then it would no longer be valine, okay? So don't look. When it's asking you what is, consider these 4 different forms of valine, which statement is true. All right. Proteins are long chains of amino acids. We know that. We know that where they're joined by peptide bonds. Which structure properly depicts the peptide bond formed between 2 generic amino acids? What you need to be able to do is identify which out of the 4 pictures are properly bonded. Okay? So remember when I said you need to learn you need to be able to know how to draw a peptide bond? You're going to have to take like this bad boy and you're going to have to add it to another one. And what I'm looking for is what's happening when we connect this, what's happening here. And then let's say another amino acid that we connect. All right. What does this bond look like? What would be there?
Doesn't it cause condensation? So 2 H's and an O leave and you'd be left with an N, a nitrogen?
Exactly. So you know that you're going to have something that's going to be like, you know, I'll just draw. I don't like that. Let's draw like, oops. This. Okay, so if we have, actually, take that back, scratch it. We know that we'll have H2N, C, and then H2N, C. So you have to count your carbons. It's always that way. Then this, like this, and then you'll have this, and then we'll go to this, and then that. Yeah, I like that. Okay. Actually,
would there be another carbon to the right of the N?
You'd have it like this. Yeah, so it'd be this, and then you would have, I think, so because this is an R group, right? So then we'd have to have another R group. And then we'd have this, like this. Yes. Right? Do we all agree? Right? That's what I was missing. So these guys, the way that like I said, the way that we like to kind of memorize things is these guys kind of will be drawn in alternate, and your R groups will be drawn in opposite. Okay, and a lot of that, again, that you don't need to know is just because of sterics. Okay, because if we had like, if this R group in red was something big and bulky, okay, and then this R group happened to be flipped on this side, then these two might interact, or this one might overpower this one, and so then this guy never actually goes to a reactive site, ever becomes reactive. Okay, so I always like to view my double bonded O's on opposites, and then my R groups opposites. Okay, but the big takeaway is what's being bonded in between, and that's this right here, because we know that we lose water. Okay, awesome. Another thing is we're going to be dealing with chirality. It's coming back. So is everyone pretty comfortable with how to determine chirality? Yes?
Yes.
Okay. I'm just going to write a generic formula. For something to be chiral, you must have 4 different things, okay? So again, you're gonna stay away from any duplicates. What I mean by that, like this, 2 H's, you're gonna stay away from double bonds. Okay? All right, let's see what else we got. Remember how we talked about histidine, phenylalanine, and things of those sorts? Those are amino acids that I would know. Phenylalanine, and I would know histidine. These, what do you need to know about them? You need to know if they're basic, acidic, polar, nonpolar. Okay, all of those. Same goes for here. You need to know those two. Why do we like phenylalanine? Because it's really, really used in chemistry. Also because this is a big medical condition, but you don't need to know that. But it's actually important. And then histidine is also one of the most abundant amino acids. It actually does pretty much everything in our body. So it's very important that you know what phenylalanine is and what histidine is in terms of just general understanding of what they are for amino acids. Okay, that's what I mean right here. Which one are they? Basic, acid, polar, nonpolar, polar, basic, etc. Okay, so you need to know their functionality in that case. Any questions on that?
So we just need to identify if they're basic, acidic, polar, or nonpolar?
Correct.
Okay.
When we are talking about the sequence of amino acids, where are those found? In what folding are those found within the protein? When we're talking about the sequence of amino acids, what are we talking about in terms of the 4 different types of protein folding?
Primary, isn't it?
What is it?
Primary.
Okay, so you need to know protein folding to just a small degree. You just need to know what's taking place in the primary, the secondary, the tertiary. Honestly, you're not ever going to really talk about quaternary, but at least these first 3 you need to know. So primary, we decided, is going to be the amino acids, right? Right? The sequencing of those. What's happening in the second level?
Hydrogen bonding.
And then specifically?
Between the backbone.
Right? But what else is happening in those, like, there's a certain terminology that we like to talk about.
It's the alpha helix or the beta pleated sheets that form.
Right. Alpha/beta sheets. Okay. That's what we need to think about. So yes, this is taking place, but really we're talking specifically about that's where our alpha and beta sheets are kind of the main topic in the secondary fold. Okay, we're forming these sheets. What's happening in the tertiary?
The intermolecular forces are making it fold in on itself.
Okay, so all I like to do for layman's terms is more folding. And then here it's like eek. Okay, because we're just like that. And then we're like this. And here we're like, okay, we're getting there. And then here we're like, right? So really you just need to know primary and secondary, just because that's where the terminology alpha beta sheets and then just sequencing of amino acids, okay? What type of force stabilizes the secondary structure of a protein? You told me that. What is it?
Bonding.
Okay, protein-protein interactions are characteristics of what?
Would that just be the quaternary, or are we still talking about structure?
Yeah, we're still talking about folding. Mm-hmm.
Okay.
So when what quaternary?
Quaternary.
Mm-hmm. Right, because there's really nothing more to do there. We've already developed everything. We've sequenced the amino acids. We've started putting them together. There's just no more that we need to worry about in the quaternary. Okay, it's at that point, it's just proteins. He's going to try and throw you for a loop about this. If we look at the general form, the general geometry of a peptide bond, what would you say that is? Think about from the first test. What is the geometry of a peptide bond?
Like a tetrahedron.
So when we're taking 2 bonds of like 2 amino acids and we're bonding them together, it's going to make a tetrahedral. Is that what you're saying? Do we agree?
Yes.
So it's going to make a tetrahedral shape. So let's draw out our peptide bond again, right? H2N, and then this, and then what's here? Our group. And then what's here? Our double bonded O, right? And then let's say that this is an N. And then what?
Another carbon in an R group.
R group, right? And then what goes here? Carbon, double bonded O, O minus. So if we're talking about just here, is that tetrahedral?
No.
So what is it?
This is supposed to be the planar one because I remember him drawing something in one
Yeah.
of his
Yep. Mm-hmm. So this is where a lot of people don't remember or like, I don't know. They just don't remember because he draws that stuff out and they're like, okay, well, I don't really care what this is. This is what we're looking at in orange. This is exactly what we're just looking at. The rest of this, I do not care. What's happening here at this particular bond between here? We know that it can't be tetrahedral. Okay, so other than that, we're left with planar. And what is planar? Planar is usually just something that's like, right? So what would we have? How many things are connected to this N?
3, so it would be the trigonal planar.
So look at this. So you have to think about this one. The thing though with the peptide bond is this planar is going to throw you a loop, because when we draw a planar, we usually just think it's like this, but this is equivalent to 180 degrees, right? Right? But here we only have one attached and one attached. And what is the degree between these guys? Even though they look drawn bent, so this is going to be on your final exam, geometry. Even though these look bent like this, they look like these are bent, reality is they're still single bonds. Here's another carbon. If I erase this right here, I'm just looking at an N and a C. I could draw that right here as N and C. Do you see this? So what is it?
Okay, so the H has nothing to do with it.
It has, it's not
planar.
It's planar. This H is so freaking weak. It doesn't do anything in terms of electronegative area or electronegativity. Okay, H's don't contribute enough to it. They're way too small. Okay, so even though these are drawn on an angle, and you think, okay, well, this looks like, Liz, this looks like a triangle. Okay, the rule with geometry for you to understand in chemistry is the substituents that are attached to it. Okay, so in planar form, you would have 3 different things, and the difference between each is 120 degrees. Linear, planar are the same thing. We have 180 degrees. It's just one substituent that you're looking at between the bonds. So even though there are 3 here, we're still just saying, okay, the distance from here to here is 180 degrees. The distance from here to here is 180 degrees. Okay, so it would be planar. Okay, any other questions on that? If you think about trigonal planar, again, that's 120 degrees. You'll have to know that for your final, at least. I'll write that here. Tetrahedral, okay. He has like 4 different things there, and the degrees is 109, all right? So, okay. And then the last question is, so I'll also trick, planar, linear, samesies, okay? Just want to let you know that. What kind of interactions are involved in the tertiary structure of a protein?
Is that just like your disulfides, your salt bridges?
Yeah. Okay. And then the next thing is he's going to ask you, okay, the area of the enzyme that interacts with the substrate. What do we call that? What's the spot where the enzyme interacts with the substrate?
Active
what is
site?
it? Yes. What is penicillin? What is it an example of?
Antibiotic.
So it is an example of an antibiotic, but what is the role of it? What does it do in the body in terms of, like, what does it act like? Think about it. Penicillin, if we're talking about inhibition, what is it like?
Like a substrate? Is that what you're looking for? Or an
No,
enzyme?
so penicillin is the antibiotic, right? That is straight up what it is. But I'm in terms of what it does to the body, how and what does it do in terms of preventing and eradicating the sickness? What does it act like?
It's a non-competitive inhibitor.
A non-competitive inhibitor. Do we agree? Yes or no?
Does it by chance act like a virus?
So penicillin is an antibiotic. What he's trying to get you at is, what is it doing to the virus to stop, or whatever you're like, because remember, penicillin, we don't really treat viruses with antibiotics. So we're treating bacteria, okay? So when you are given penicillin, what is it doing to the bacteria? What is it trying to do to the bacteria? It's not just killing them, but what is it trying to do?
Break them apart.
Right. It's trying to prevent from more bacteria growing. So if we think about inhibitors and what inhibitors do, what can we say penicillin resembles?
An inhibitor?
It is an inhibitor. What kind of inhibitor? Stupid mouse, dude.
Would it be a competitive one?
So if your options are irreversible, non-competitive, allosteric, or reversible, what would you say?
Irreversible?
I would say allosteric, because then you'd be changing the bacteria, or I'm not exactly sure, but that's just what I thought.
Let's think about it. So a reversible inhibitor, we know it can't be that, right? Because why would, if we're trying to use penicillin to eradicate bacteria, why would we want it to be a reversible inhibitor? That, it doesn't make sense, because that means it can come back, okay? So we know it can't be that. Non-competitive inhibitor, well, if you have a bacterial situation and you're giving it, you're trying to put penicillin in the body that's suffering from bacteria. Non-competitive, remember how I said, is just like whenever it might just happen. So can we say that it would be a non-competitive inhibitor?
No.
Okay, so now right, so now our only options are in irreversible, or allosteric? Remember what allosterics do. They go to the allosteric site and just change the substrate spot, okay? Change the way that it looks. So it'd be from a triangle to a square. So if penicillin just goes to the non-active site and then changed it, what if our bacteria mutated? What if our bacteria evolved and now we had some square bacteria and some triangle bacteria? What's going to happen? The triangle bacteria are going to die, but then my square bacteria are just going to live, evolve. And so then what's the point of penicillin there? So penicillin acts as what?
It's the irreversible inhibitor.
Exactly. It's an irreversible inhibitor, okay? Because it's going to inhibit the active site. It's going to inhibit that enzyme from working, okay? Or that bacteria from working. That's what we want, okay? So you don't want to push a drug into an allosteric site because your bacteria could evolve, or whatever it is that you're doing could start to make more of that, okay? We need it to stop. We need it to go the opposite way, act differently. Okay, so we want it to be an irreversible. The area, and then what else? Then the rest of it is just going to be your questions. So please choose blah, 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 and those are all personal. They're not worth points. That's the exam. That is straight up the exam. On this exam, there might be 2 or 3 questions that are going to cause you some havoc. So I'm going to give you some tips right now, if my mouse will work. First thing that I would suggest, I think that you should re-look at the exam. I want you to, actually, I'm not just suggesting, I highly recommend, if you haven't already, go to the exam 4 folder and look at the answers as well as take that practice exam, okay? It's literally like maybe 20, 30, I don't know. It's really, really fast. Here's why. 1, pictures are on there that are gonna be here, okay? Your pictures are there. It's gonna show you, it's gonna ask you for those chiral carbons. It's gonna ask you to show the peptide bonds. He pulls from those because most of the people most of the time don't actually go on those and look. So look there, begin there. Second, okay? You need to be able to, on there it's gonna have a couple of things talking about heavy metals, all right? So Pb2 plus, maybe mercury, something like that. I highly recommend that you look at that question, all right? Because that might also be here on this exam. If not, reference it, all right? In the sense of, what do heavy metals do? Okay, what do they contribute? What do they disrupt? How do they disrupt? Okay, and that's about it. And then the last thing would just be looking up what the coronavirus vaccines are. What do they do? We talked about that. Targets and deals with the mRNA, okay? So those are the things that I would just say are kind of the softballs for this exam, just because it's like, where did that come from? But I think too, though, he did mention a little bit on, there was some previous readings on the files page about coronavirus and some vaccines and stuff, but again, nobody really goes in and digs. So that's the tip that I'm going to tell you for that. Otherwise, this is the exam, and it's really not that bad. Okay, you don't need to know a lot of specifics. I'd say the most specifics really stem in this primary, secondary, tertiary, quaternary. What's happening in each of those levels? All right, in terms of quaternary, protein. Tertiary, we have our disulfide salt bridges. Why? Because those are the heavier, the heavier bonds, the stronger bonds, which makes sense because now we're in the 3rd level of protein folding, right? It's not just the simple tiny bonds anymore. It makes sense. We're getting tighter. Okay, secondary, you need to know just the terminology of alpha beta sheets, all right? And then, and hydrogen bonding, which makes sense. Again, now we're at the 2nd level. We know hydrogen bonding is strong, okay? So it just keeps building that way. And then we have to begin somewhere primary. It's just our sequence, okay? This geometry, planar, linear, are the same, so don't get swayed by that. Remember those two amino acids. You will have to identify how many chiral carbons are in a structure, and that's it, okay? Any questions, comments, or concerns? Now, I will say, I think number 3, I think the third or fourth question, the one that's talking about the inhibitors, it says it does not bind to the active site. I'm gonna have to, everyone that came here today is pretty much giving that free point, but it is an allosteric site, but I think what he's looking for is non-competitive, okay? So for that question, please don't put anything other than non-competitive in there, all right? And even if you get it wrong, we'll fix it. I'll probably just end up knocking that question out of the point total, okay? Any other questions, comments, concerns, worries?
Can you repeat what you said about mercury and heavy metals? I missed that.
Yeah, so you'll want to know, you'll want to know about what is it that those heavy metals do in terms of their, when they interact with other things. So heavy metals have those extra ions, okay? And the key takeaway is that